Good afternoon, everybody. Welcome back to the joint RUMA and SFAM meeting this year. This is the last session in the series, and it's on innovations in animal health. I think it's really important that we're equipping farmers and vets with the tools and solutions to keep farm animals in good health and ensure sustainable and efficient farming practice. Innovations such as rapid diagnostics for early detection of new therapeutics for treating infections will support and promote targeted animal health management and contribute to the prevention of disease where necessary and treating infections accurately where we can. We have the opportunity this afternoon to hear from 5 different presenters this morning on and around the subject of innovation in agriculture, especially in relation to AMR and diagnostics. The detailed biographies of each presenter are actually on the conference website, so I won't go through them in detail. So firstly, we will hear from Professor Conrad Lichtenstein talking about DNA therapeutics administered before antibiotics to inactivate the resistance of bacterial pathogens. The next to present will be Dr. Fiona Lovatt, who is a sheep veterinary consultant for the Flock Health Club. Dr. Lovatt undertook a PhD at the University of Durham while still practicing as a vet, so that's pretty amazing. Dr. Lovatt is the past president of the Sheep Veterinary Society and currently chairs the UK Sheep Antibiotic Guardian Group. Next, we have Professor Henry Buller. Henry describes himself as a professor of more than human geography at the University of Exeter. Professor Buller will talk about the DIAL project, the potential for rapid diagnostics in treating livestock diseases. We will then hear from Dr. Salvatore Galgano, from the Monogastric Science Research Group at the SRUC. To close off the session, the presentation session, we'll be hearing from Professor Jasmeet Kaler. Jasmeet is a professor in epidemiology and precision livestock informatics at Nottingham University, and she will talk about the use of imaging and sensor technologies for the early detection of disease. So following these presentations, we'll all come back together for a panel session, receive questions from the audience. This will be used using the chat function, and you'll see this at the right hand of the screen. So with that, I will stop whittering on and let us listen to the presentations which are coming to you now. So I'll see you for the panel session at the end. Thank you.
I'd like to thank the organisers for inviting me to participate in this conference, and also to thank my co-founders Frank Massam and Guy Mikawa, and our research officers Hannah Patrick, Colin Jackson and Eleanor Jones. So the primary problem with the traditional response to the emergence of antibiotic resistance is that, as you know, bacteria very, very quickly become resistant. And the problem is we're developing small molecules, which is expensive and slow, clinical trials, and it's kind of like hardware. And the bacteria respond with a software update. They either acquire an antibiotic resistance gene from another bacterium, or they evolve one from existing genes that can degrade antibiotics. And so when we try and fight that by generating new antibiotics, perhaps ones that have been tweaked, they very quickly come up with a new software solution. And it's really the classic Red Queen race where you have to keep running to stay in the same place. And so the big question then is, how can we run twice as fast as bacteria? And the technology that we've been developing essentially uses the gifts that Mother Nature has provided. And the first big gift was the discovery of restriction enzymes and the birth of recombinant DNA technology in the 1970s. And then in the 2010s, another immune response that bacteria have evolved to fight off phage infection, bacteriophage infection, is the adaptive immunity of CRISPR-Cas, which essentially is a blind endonuclease that cannot cut DNA, cannot recognize, can cut DNA, but doesn't know where to cut. and it's guided there by a guide RNA. And so the wonderful thing about this is that you can change the specificity of that complex, that ribonucleoprotein complex, simply by changing the RNA sequence. And so we've adapted this endogenous adaptive immune response that bacteria have back on bacteria themselves by engineering RNA-guided nucleases that will attack antibiotic resistance genes. And so essentially we now have a software approach to combating the software response that bacteria have. And in that way we can resurrect existing antibiotics. And I'm going to show you today some of the work we've been doing, for example, to inactivate beta-lactamase genes, first of all the TEM beta-lactamase, and then show that we can inactivate it with a multifunctional construct encoding many guide RNAs. We can inactivate many different families of beta-lactamase genes. One's not restricted to beta-lactamase genes. One can knock out virulence genes. One can knock out other antibiotic resistance genes. The technology is agnostic. It just wants a DNA sequence and for you to engineer a specificity to it. But of course the challenge is we have to be able to get that DNA into the bacteria. Because what we've developed essentially is a companion DNA therapeutic to use together with antibiotics. And we've developed what we call our transmids, which are essentially conjugative plasmids that are encapsulated in a bacteriophage coat. So we can deliver directly by phage infection, but also either subsequently by bacterial conjugation or directly by bacterial conjugation if we use a commensal bacterial probiotic donor to deliver what we call symbiotics to the target bacteria, which might be bacteria in the gut microbiome, for example. And we validated this technology in mouse model studies, both for the treatment of infection, therapeutically, in the peritoneum, and also prophylactically to clear the gut microbiome. And so our technology can be used to allow existing antibiotics to work again and to protect new antibiotics from the emergence of resistance. An important point is that we avoid direct killing by RNA-induced DNA damage. Typically, Cas9 introduces double-strand breaks in DNA, and if that target site's in the chromosome will suffer that damage and the cell will die. And if it's on a plasmid, that plasmid may have a post-segregational killing mechanism. So if the plasmid's lost, the plasmid kills the cell. And we've taken a variety of approaches to do that, which I don't really have time to go into today, but we'll talk about a little bit more later. And perhaps of most relevance to this particular conference are the veterinary applications of our technology to limit the spread of antibiotic resistance genes into the bacterial gene pool and to reduce the transmission of antibiotic resistance from animals to humans. And we think, although we haven't begun these studies yet, but we're planning to, that the pig gut microbiome can serve as a proxy for evaluating the clearing of the human gut microbiome of such antibiotic resistance genes, and therefore in humans would protect patients in high-risk environments from opportunistic infections, for example, urinary tract infections, and also perhaps in the anticipation of surgery, for example, a hip replacement, where if they were to have an opportunistic infection, it would be treatable with an antibiotic. A construct we made initially, which is expressed on a small plasmid. The plasmid carries chloramphenicol resistance as a marker. We have the Cas9 endonuclease, the tracer RNA, and a region where we can put in our guide RNA sequence. And the guide RNA transcript plus the tracer RNA assemble to produce the processed product, which is the mature guide RNA that then assembles with the Cas9. And we identified a target sequence present in a number of different genes of the BLA-TEM family that's well conserved, so that it should knock out all of the following genes shown in the slide. And as a negative control, we built in an isogenic construct with Cas9, identical to the one shown here, but simply lacking the 10 spacer. Slide 6 shows some assays that we developed to demonstrate activity. The first was a plasmid transformation assay, where you take a cell culture that carries the beta-lactamase gene, transform them with our construct, select for chloramphenicol resistance, pick colonies, and then screen them for sensitivity to carbenicillin. And so this shows you replica plating toothpick assay, where on the right-hand side of panel at the top left, you can see an activation of carbenicillin successfully with our construct, but not with the one lacking the spacer. We also did a conjugation assay where we mated in a beta-lactamase gene into a recipient cell carrying our construct, and we got a 20,000-fold reduction in mating efficiency because the incoming plasmid carbenicillin resistance gene was inactivated by the resident plasmid. And in the third assay, this was done by phage infection and using nitrocephin, which is a chromogenic substrate of beta-lactamase, we were able to show that the supernatant culture, because the enzyme secreted, of cells that had been infected by the phage had reduced beta-lactamase activity. We then built a multiplex construct where we expressed 8 different guide RNAs designed to target conserved regions of members of 8 different beta-lactamase families. And then the following slide shows our strategy, where what we then did is source members of each of those 8 families, cloned their beta-lactamase gene into our plasmid vector, introduced that into 8 different isogenic E. coli strains, to which we then subsequently introduced by transformation our plasmid encoding the Cas9 plus the 8 guide RNAs, and then did the similar plasmid transformation assay and screen for antibiotic inactivation that I described previously. And we showed that they all work. And in fact, that's interesting because presumably there's some dilution in that you're making essentially 8 different ribonuclear protein complexes targeting 8 different beta-lactamase genes, yet each one is sufficient to inactivate efficiently the target antibiotic resistance gene that we've introduced in each of our 8 independent assays. But of course, we actually have to have a way of delivering our transmids, and I mentioned earlier what we've done is developed a dual delivery system. So if we encapsulate our transmid in a phage coat, which we do in vitro packaging, by induction of a prophage that itself can't be packaged, but can package our resident transmid. If we produce that, purify it, concentrate it, and use that as our reagent, that's able to deliver the transmid by infection to our target bacteria, or alternatively, as I mentioned earlier, we can also deliver directly from a gut commensal by bacterial conjugation. If we do it by what we call transmeduction, infection in a phage lambda packaged transmid, for example, we took 2 different cell lines, one carrying the beta-lactamase gene from Klebsiella pneumonia carbenamase 3, which isn't a target for our construct because we only put in 1 guide RNA here, targeting TEM. You can see that you can select for successful infection on kanamycin plates in this case, but if you screen on plates containing carbenicillin, you get a massive reduction in cells able to grow because they've been inactivated. We then went on to do an animal model study. First of all, we did some therapeutic studies by intraperitoneal injection of bacteria into mice, followed by the injection of our phage lambda packaged transmid. And then we, at 6 and 24 hours, sacrificed the mice, performed peritoneal washes, and assayed the mice on appropriate drug plates to evaluate efficiency of transmid delivery and efficiency of carbenicillin resistance inactivation by virtue of activation of the 10 beta-lactamase. And we had a control experiment, isogenic strain carrying not the beta-lactamase 10 gene, but the beta-lactamase KPC3 gene. Here you'd expect successful delivery, but failure to inactivate the target gene, and so the bacteria should remain carbapenicillin resistant. We were able to obtain, in this case, our transmit carried a tetracycline resistance marker, but we were able to obtain, with just a single injection of the transmit, around 85% efficiency of delivery to the bacteria in the peritoneum, and in all those bacteria carrying the transmit, over 99% efficiency of inactivation. Next slide shows our animal model studies in the gut microbiome. And here what we did was feed mice an E. coli recipient strain carrying the beta-lactamase gene, either the 10-beta-lactamase, the experiment, or the KPC3 beta-lactamase, the control. The mice were then fed streptomycin in their drinking water to kill off their endogenous E. coli, and within very quickly, the introduced E. coli strain colonized the mouse gut microbiome so that we could recover high concentrations of bacteria from the feces. And at the beginning of the second week, we then fed the donor strain, an isogenic strain, but carrying the transmit, and then looked for successful conjugation of the transmit to the recipients in the mouse gut and consequent inactivation of the target BLA10 beta-lactamase gene. And the study was essentially modeled on one developed by Karen Krogfeldt in Copenhagen and her lab. And we used sub-inhibitory tetracycline concentrations, which she'd used to demonstrate efficient spread of a plasmid, and we'd shown to demonstrate efficient spread of our transmit with its consequence inactivation. Where really within a couple of days, we got close to 100% efficiency of conjugation. And in those bacteria that had received the transmit by conjugation, over the 99% efficiency of inactivation. Well, we're now working on developing a proof of concept study in a collaboration with the Japanese pharmaceutical company for lung infections. But we are also developing the technology for applications for clearing the microbiome. And so what I've shown you today is that we can inactivate antibiotic resistance genes efficiently. We can multiplex to inactivate multiple different antibiotic resistance genes. And we can also use it to remove antibiotic resistance genes from the microbiome. And now we're planning on doing some pig gut microbiome studies to develop the technology further. Thank you very much.
Hi, so I'm Fiona Lovatt and I'm the clinical lead for Farm Vet Champions, which I'll explain about as we go through. So starting on a slide that, a picture that probably is familiar to a number of people in the audience, and it's from the O'Neill report that was published in 2016, and shocking statistic that in the next 30 years, AMR will be responsible for 10 million global human deaths. And so obviously from a One Health point of view, you know, that's no surprise to this audience. But in a veterinary point of view, I'm a sheep vet myself. And this data was published a couple of years ago looking at sheep flocks, and they took the susceptibilities testing of E. coli recovered during the lambing season. And plenty of occasions, nothing to worry about, but there were two farms particular there, in fact, more than two, but Farm 4 and Farm 9, where we have really widespread multi-resistant E. coli in those lambing flocks. So the reason I got involved initially as a sheep vet in antimicrobial stewardship is it's not just that I'm concerned about from a human health point of view, I want to know that my kids can go into hospital and not be scared of a routine procedure. I also want to know that my farming neighbour can treat a sick animal when necessary, and that he is using antibiotics responsibly. Now, we have made enormous progress within the livestock sectors in the UK. I've been working with RUMA since 2016 as the sheep vet representative. We've in food producing animals in the UK, we have demonstrated a 45% reduction in total use of antibiotics in farm animals. And we, as a veterinary profession, are extremely proud of the progress we've made. But it has to be said that we've largely been able to evidence this in poultry and pig production, where the data is easier to come by. So every sector is different. So we have on the left-hand side of this graph, the more integrated sectors, where we have very few vets working in poultry or fish, very closely aligned sectors, few producers, large units, small number of vets to get to, and relatively easy to collect data from those sort of enterprises. On the other side of the picture, we have the ruminant sectors. We've got 70,000 beef and sheep farms in the UK, small, extensive units, many producers, often family farms working with small general practitioner vets, loads of vets, and loads of farmers, and often challenges with data. So, and we may have farms where it's difficult for them to collect data on an animal specific basis. They may have a couple of thousand sheep or several hundred beef cattle, and individual data collection is not always well recorded. So in the RUMA targets, we looked at these issues within the ruminant sectors and sort of teased it out into the importance of capturing and using data well, good veterinary farmer engagement, and primarily, and what we're all really driving for, is good health and welfare. So we know that if we've got happy, healthy animals, there is no need to turn to antibiotics. That's that holistic aim of us all, is to have healthy animals that are well fed, good welfare, and then we can sort of drive responsible use of medicines from that. So, but because of these challenges, we had a job to do, and we really wanted to capture vets at the point of them having a conversation on farm. So general practitioner, farm vets, getting out of a car at a milking parlour or in a lambing shed, pulling on wellies and thinking, what can I do? How can I have this discussion with this client? And can we make progress? So and that's where we launched the Farm Vet Champions. So it's a massive veterinary collaboration across all UK livestock sectors. It's spearheaded by RCVS Knowledge, a charity, and it's funded by the VMD, the Veterinary Medicine Directive. And each of these logos here represents the organisations who we've got engaged. They're members of our steering group. They're really there in the driving, have been there right from the start. And we were delivering it in 2 phases, and phase 1 is up and running. So phase 1 is basically about 20 plus hours of CPD that's specifically targeted towards general practitioner farm vets working in UK farm animal practice, but it's free to access to all veterinary professionals or vet students, and also to practice support staff. So sometimes in a farm animal practice, actually the receptionist or the vet tech, or there's somebody else who is an important member of that team in the prescription and use of antibiotics, although all antibiotics must go through a vet as the prescriber. And this phase 1 CPD, it's basically into 3 sections. We've got the introduction to medicines, absolute essential need to know information on the use of antibiotics, on the legalities of prescribing veterinary medicines, on the One Health aspects, on the development of AMR. just important stuff that all of us were taught at some stage in our veterinary career, but absolutely all of us could also do with reminding. There's a really important and quite exciting section on farmer and vet behaviour, attitudes and communications. So it's all very well for a vet to be technically very competent, but if they're not able to communicate that with their farmer clients, then they can't progress. So that's key, and we've got a number of different expert speakers, just small bite-size bits of attitude and communication training with a view to signposting vets to further information if you know it piques their interest. And then most importantly, we've got the species specific modules. So we've got, for example, in cattle, we've got a module on dairy cows, a module on suckler cows, and a module on young stock calf species specific stuff, all delivered by the species specialist associations. But the thread right through it, it's absolutely not, okay, so Mrs. Vet, here, Mr. Vet, here you are, what antibiotic do you use for this situation in this turkey or in this calf? It's much more holistic like that. So we've kind of gone way back and said, how do we ensure healthy animals? So can we encourage vets and farmers to properly plan ahead? So make sure the diet's right, make sure they're using records appropriately, make sure there is a good farmer veterinary relationship. That's a plan stage. And then prevent unnecessary disease. So that might be good shelter, it might be appropriate environment, good living conditions, nice, dry, clean bedding, well ventilated, hygienic situation in the lambing shed, and then protect is actually protecting that flock or herd. So in terms of a neonate or baby animal, that's making sure they get good quality colostrum. So make sure that the mothers are well fed, in good body condition, and can produce decent colostrum, so that the lamb or the calf or the piglet has a good quantity of immunoglobulins as they take in the colostrum, so they get enough, it's good enough quality, and they get it quickly enough. And then in terms of the rest of the herd or flock, it's actually, are we using appropriate vaccinations, of which you know, there's not every single vaccine available for cattle or sheep or whatever is appropriate on each different farm, but through the vet conversations and through risk assessment, which vaccinations would make a difference, and how can we make that flock or herd as healthy as possible. I'm just going to go for a specific example, and you'll have to excuse me, as a sheep vet, I'll have to go back to sheep. So in communicating with sheep farmers, historically, a large number of sheep farmers had treated many lambs prophylactically at birth with antibiotics. And the reason farmers were doing that is because at some stage, their vet had told them that was the most appropriate way to treat that disease. So they were acting on what was good veterinary advice at the time. But it's hard. They believe they're doing the right thing, and they believe they're preserving animal welfare. But as I go back to this slide that I had right at the beginning of levels of resistance we've got in E. coli on some sheep farms, you can see we have got a real issue. And it's something that as a profession and as a sheep farming industry, we have been working very hard in the last few years to communicate to farmers. So there on one hand, we've got a sheep farmer who absolutely wants the best for his lambs, and he's convinced that he may compromise welfare if he doesn't use routine antibiotics. So then we have a challenge, and the vet conversation is really important. And the way we work through that with planning ahead, preventing, making sure shelter and buildings are hygienic and appropriate, and actually protecting that flock. So we'll make sure that the ewes are fit and well fed so that they can produce good quality colostrum and fill the lamb with colostrum, and there's no need to turn to antibiotics. But it's not something that we can just change overnight. So I spend a lot of time talking through this with vets, and we've done this in the training for the Farm Vet Champions. Can we divide the flock? Can we identify a lamb that's at less risk, that's absolutely certainly doesn't need any antibiotic, but can we actually target those really high risk lambs and help farmers as they're making that change from a situation where they thought it was normal to treat every lamb to a situation where they're very targeted about it, and we can use treatments appropriately. And it's an active plan. It's not a one size fits all, but we're taking people on a journey, and we're all the time ensuring good welfare as well. So it's kind of just as a case report, I suppose, but encouraging vets to be out on farm planning ahead. A vet is an expensive thing to have on a farm. Economics in the sheep industry are really, really tight. So we have a challenge there, and convincing hard to change farmers that actually there are better ways of doing things is all part of that's where the communication attitudes all come into it. So it's a massive work, and I've only sort of described the small aspect of lambs in a sheep flock, but you can imagine we've got this similar issues on cattle farms and dairy farms, beef supply units, and we've also got modules within the Farm Vet Champions on game birds, on turkey rearing, on laying hens. They're all things that a vet in practice may have to turn their hand to. They may not be the expert in that, but they're the vet on call, and the farmers come to them and they need to know what is best practice and how can we absolutely ensure responsible use of medicines. And it's a good story to tell. So we have been tracking sales of oral antibiotics in UK lambs, and in the last 5 years we've reduced actual use of antibiotics by 34%. So we can track good progress. It's not something that will change overnight, but we're making progress, and that's great to see. But just quickly talk about phase 2 of the Farm Vet Champions, because delivering CPD is one thing to vets, but that's a bit sort of one way. Actually, I think this phase 2 is kind of much more exciting. It's about setting SMART goals and building a real community and network of Farm Vet Champions. So we've gone back to where Ruma identified important areas of needing to engage, needing to capture data, and needing to ensure responsible use and good health and welfare. And then we've said as we set this up, and it's literally in progress, we'll set up the platform that as a vet says, okay, for my practice or in my situation, as I talk to my farm clients, I'm going to set, I'm going to speak to 20 sheep farmer clients by the end of November, so well in time for lambing, about sorting out their nutrition, ensuring that they've got good protocols in place, and working specifically on more responsible use for their next lambing season. And as the vet can set that SMART goal and track it on the platform will come back to them at the end of November and say, you set this goal, how are you doing with that? And the vet will have an option to say, oh yes, I've achieved that, and here's the evidence, or yes, I've got there, or maybe I'm modifying that goal and this is how I'm changing it. But it will actually be interactive to a way that we can encourage vets to be very smart, so specific, with measurable goals are achievable and within a really realistic time frame, and actually that interaction as well as a community of farm vets. So it's a very isolating job being a farm vet out there with your clients who are small businesses in a remote area. We all need encouragement to do the right thing and to be, we're all at different places on the journey, and we have some amazing examples of really good practice on farms and what vets are doing. But we also have people who we need to pull along a bit, and they may be a bit set in their ways, or they maybe could have a bit of encouragement. So that's really where we are with the Farm Vet Champions. We've got 5,500 farm vets in the UK, and we're quite ambitious. We want to reach all of them. Actually, half of them would be good for starters. But if you're listening and you're a vet or you've got contact with vets, then just please encourage them to join us. This is something that, you know, we need to take everyone with us. So as vets, we are responsible for the use of antibiotics in animals. We are the guardians of those antibiotics, but we have a massive job to do. It's a huge responsibility, and we can make that difference together. But it really is going to be at the level of every single consultation, every single conversation with a farmer within a practice. You know, it's like one slice at a time. So yeah, anyone who's listening and can share that, spread the word, please do.
Good afternoon. I'm a social scientist at the University of Exeter, working primarily in farm animal health and welfare, and most recently in the area of antimicrobial use and the diagnostic and treatment decision practices that accompany that use. One research project that I'm currently leading is looking specifically at the role and place of new diagnostic and animal surveillance technologies in informing the prescription of antimicrobials and the treatment of livestock. Although that project is still very much ongoing, we've published several papers from it already, specifically on new pen-side and rapid diagnostic tests in farm animal clinical veterinary practice, and on diagnostic technology development pathways, both of which I'll draw upon in this short talk. So in this talk, I want to report on three aspects of our research. First, the manner in which new rapid pen-side and point-of-care diagnostic test technologies impact, or perhaps don't impact, upon clinical practice and ultimately upon antimicrobial use in livestock systems. Second, I want to look briefly at some of the barriers impeding diagnostic developers from negotiating the complex professional and commercial landscape of new veterinary diagnostic technologies, bringing them into adoption and use. And third, I want to mention some of the alternative surveillance and monitoring technologies that are emerging in parallel as an important additional component to animal health care and some of the challenges that these bring. As I said a moment ago, this comes primarily from a current research project which is called DIAL, Diagnostic Innovation in Livestock, towards more effective and sustainable applications of antibiotics in livestock farming, which, and this is very much the joy of this kind of research, is really a truly collaborative interdisciplinary team of researchers drawing upon veterinary science, epidemiology, social science, technological studies, and so on. As part of that project, we've carried out a survey of some 200 farm animal veterinarians. We've interviewed 30 cattle, pig and poultry veterinarians and the same number of farmers in those three sectors. We've held focus groups with members of veterinary companies, have interviewed around 30 diagnostic development companies across the UK, and we've also held workshops in London with different diagnostic development people and are planning further workshops in the coming months. So let me turn to the first of my three themes. As I'm sure everyone here is only too aware, the last decade has seen a distinctive push for new and innovative diagnostic technologies in both human and animal care as one critical means of reducing unnecessary antimicrobial use. Part of the emphasis here in both the medical and the veterinary sectors has been on rapid point of care and in farm animal veterinary practice pen-side diagnostic tests. The argument being that more accurate and speedy testing will allow for more accurate treatment, avoid precipitous, avoidable and unnecessary antimicrobial use, and thus reduce the potential for resistance. A corollary to this emphasis on new rapid diagnostic technologies has sometimes been an implicit or even explicit criticism of more traditional and long-standing diagnostic practices and behaviours. A report by the UK Review on Antimicrobial Resistance, chaired by Jim O'Neill, for example, published in 2015, makes the following point, and I quote, When doctors, and here we might add veterinarians, decide whether to prescribe an antibiotic, they usually use so-called empirical diagnosis. They will use their expertise, intuition and professional judgment to guess whether an infection is present and what is likely to be causing it, and thus the most appropriate treatment. In some instances, diagnostic tools are used later to confirm or change that prescription. This is a process that has remained basically unchanged in decades. The solution for the O'Neill Committee, and indeed others, lies principally in rapid diagnostic technology. I quote again from their 2015 report, "Rapid diagnostic tools for bacterial infections which allow doctors," and here again we might add veterinarians, "to identify the nature of an infection in minutes instead of hours or days, have the potential to transform the diagnosis and treatment process from an empirical one to a precise one." At 6 or 7 years on then, we might ask what has been the impact of this drive towards rapid diagnostic tests on veterinary diagnostic practice. Are these precise processes replacing empirical guesswork, as the O'Neill report would have it? And are they helping to achieve more appropriate antimicrobial use? So I want to try and answer each of those questions in turn. Firstly, are rapid diagnostic tests making a difference in veterinary practice? There is an increasing number of rapid pen-side or cow-side point-of-care tests on the market at the moment, and they're certainly becoming increasingly common in clinical veterinary practice. They produce fast results that allow prescriptions and treatment decisions to be made quickly, accurately, and often at lower cost. Moreover, they allow veterinarians to reassure farmers that the decision to treat or not to treat is justified. In our survey and from our interviews with veterinarians, we observe, of course, the growing use of scour test devices, ketone tests, glutavac tests, the California milk test, BVD SNAP testing, calcium testing devices, and even portable PCR and ELISA tests. Many veterinary practices train their dairy clients in the home use of culture plates to identify mastitis and other infections. Overall, pen-side and rapid tests are certainly being increasingly used within the profession. However, there are important sector differences. We find that it is mainly in the dairy sector that such technologies have found a ready market, far more so than, say, in the pig and poultry sectors. Nevertheless, the more general adoption of rapid tests has often been limited by a number of factors, including the fact that many vets express concerns over the practical use of rapid tests in on-farm environments. They are concerned about the limited range of disease applications that the tests currently on the market have, and by the fact that for many cattle veterinarians, these tests rarely offer unequivocal confirmation of a specific pathogen on which to base a prescription or treatment decision. And finally, conventional lab-based diagnostic tests are becoming faster and more accessible. They are rapid, they may not be pen-side yet, though I have no doubt that that is coming. Indeed, perceptions of the accuracy of rapid and point-of-care tests was a theme that emerged repeatedly in our survey, with several veterinarians maintaining that manufacturers' sensitivity and specificity claims were not always easy to ascertain or confirm for some of the rapid tests in current use. One cattle veterinarian put it like this: "I'm not sure of the sensitivity of those things, so even in the absence of a bacterial positive, I'm not sure we'd always be brave enough to say we're not going to treat on that." I think the rapidity depends on that aspect of it. If they don't have a sensitivity that's as good as the labs, or nearly as good, then they're probably not going to be used. The poultry and pig sectors, where post-mortem diagnostic tests on the one hand and vaccination on the other are a lot more common, have not seen this significant rise in the use of these rapid tests, though some could certainly envisage greater use if the technology and the usability was there. Here's a pig veterinarian that we interviewed. If you could plop a drop of scour on a plate and it says, yes, it's an E. coli, yes, it has virulence factors to be significant, and this is the resistance profile, then that would allow you to institute treatment faster and more logically. My second question: are rapid tests replacing more traditional empirical approaches to diagnosis? The answer here is clearly no. The empirical approach to diagnosis, guesswork as the O'Neill report had it, based on experience, observation and clinical judgment, remains very resilient and strongly defended by experienced clinicians. Prescription and treatment decisions continue to be made with a high degree of diagnostic confidence, even in the absence of a specific test, drawing upon established diagnostic procedures, knowledge of the farm, herd health, and in many cases, past experience in dealing with specific disease profiles, particularly routine production diseases. So what we're not seeing in practice is the abandonment of these more traditional forms of professional expertise in health and welfare assessment in favour of a supposedly more precise scientific approach to pathogen detection through specific tests. That being said, what we are seeing, particularly, is the use of culture plates in dairy systems, is the diversification of responsibility for disease testing, not so much away from veterinarians, but increasingly to include farmers, particularly those trained in or with experience in interpreting culture plate results and acting accordingly. So my third question is whether the growing number of pen-side diagnostic tests is contributing to reductions in antimicrobial use. Of course, this is the most difficult one to answer. Recent years have certainly seen significant reductions in the volumes of antimicrobial medicines used in the pig, poultry and dairy sectors in the UK, and stewardship programmes are certainly being widely hailed as successful in this goal. The problem is that most of the more commonly used pen-side and point-of-care tests currently in use are not really relevant to the detection of specific bacterial infections and to subsequent antimicrobial treatments, despite the very clear demand that exists for exactly that kind of rapid test. Although veterinarians and farmers are often concerned about the time it takes to get test results back from the laboratories, which can drive the use of antimicrobials before the results do come back, in this way, antimicrobials act to a degree as their own diagnosis process. Rapid tests are considered less accurate, less detailed or sensitive enough upon which to treat with antimicrobials, thereby obliging the veterinarian to rely upon clinical empirical experience or indeed another round of further testing. Overall, therefore, veterinarians in our research saw the future of pen-side devices not in specifically reducing unnecessary antimicrobial use, but as part of an extended diagnostic pathway that might begin with a simple rule in or rule out using rapid testing and then lead to a more investigative testing or treatment. A positive result, it's easy, isn't it? It's the negative ones that are difficult, as one cattle veterinarian put it. A single rapid test, then, is unlikely to be considered definitive. In practice, building up to a clinical decision requires the mobilisation of different and multiple forms of evidence. Rapid tests may be useful to this, but they are unlikely to replace time-deepened practices of knowing through experience and evidence building. So while the relative simplifications inherent in rapid and point-of-care diagnostic tests might offer a considerable advantage in speed and potential pathogen targeting, the complexity of on-farm biotic environments mitigates against their exclusive solitary use. At the end of the day, one might suggest that it is in the ability to distinguish and select amongst complex microbial environments of farms that the broader empirical diagnostic practice reveals its greater value. Any simplification of scientific diagnostic processes may well have a localized impact on reducing some erroneous prescription and treatment decisions, but may do little ultimately, if anything, to manage the wider biotic complexity of the contemporary farm. Coming back now to the three aspects of our research that I wanted to talk about, we might ask, given the policy push towards rapid diagnostic technologies and the perceived value tests could have in identifying and treating pathogens, why do there seem to be relatively few new diagnostic tests used in clinical veterinary practice in this way? There is a clear demand amongst veterinarians for better, faster, more accurate diagnostic tests that can identify and distinguish bacterial pathogens, that can indicate sensitivity and likely resistance to specific antibiotics, that can allow for the pinpointing of targeting treatments. So why isn't it happening? The work we've been undertaking with diagnostic developers points to a persistent disconnect between the veterinary profession on the one hand and the diagnostic development sector on the other, itself often far better aligned with the medical community than with veterinarians. Perhaps this mediated push towards rapid technologies and the implicit critique of traditional empirical practices in veterinary diagnosis that has accompanied it has exacerbated further this disconnect. Perhaps, too, veterinary diagnosis is seen as a black box disconnected from broader commercial food chain concerns and value systems. We note with interest how in the UK the Red Tractor food product certification scheme, for example, now requires a mandatory veterinarian-led diagnostic or sensitivity test for the use of all high-priority critically important antibiotics on Red Tractor farms. Here then is an interesting link between the need for effective technology and broader food chain valuation and regulation. Maybe these kinds of linkages will provide greater stimulus to the development of the sort of pen-side and point-of-care diagnostic tests that veterinarians are asking for. But, and I will end with this, there is another area of technological innovation that in some ways offers a parallel and complementary perspective on addressing animal health, which may ultimately prove to be just as important as rapid diagnostic technology. The new digital and precision monitoring and animal health surveillance technologies are rapidly moving forward, allowing for individual and herd or flock real-time monitoring, in vivo data gathering, psychological and sociological behavioural indicators remotely recorded, and so on. As one farmer said to us recently, quote, "I've got this cow manager, a tag system, which is a massive help in spotting any sick cows. Basically, it monitors their temperature and their eating behaviour and basically tells you 48 hours before anything goes wrong with the cow and gives you the opportunity to get in there. And that's been a big thing in our antibiotic reduction. That's been a big factor in reducing our antibiotic use." So in this project, we're looking at this more closely as another component in the veterinarians and the farmers' armoury for dealing with animal health and disease. And I think it also raises a number of important questions. To what extent can these new monitoring technologies bring new and additional early information on animal health and disease that are not immediately being picked up by more traditional approaches? What alert mechanisms can be put in place, and what are the points of intervention for the farmer or stock person? Who owns the data? How can it be shared and transmitted, and between whom? And finally, what impact might these new technologies have on conventional forms of stock person or veterinarian observation of the animals in their care? So, to conclude, there's a lot going on in these new technological developments, and this has the potential to have a significant impact not only upon existing approaches to disease diagnosis and to the conventional observational, empirical and care relationships between stock persons and animals, but also and critically upon how and when antimicrobials are used on livestock farms. Thank you.
Good morning, everyone. My name is Salvatore Galgano, and I would like to thank the Society for Applied Microbiology and RUMA for inviting us today to talk about our project, which focuses on the potential use of peracetic acid nanoparticles for non-antibiotic modulation of gut microbiota in broilers and possible modulation of performance. I must say, it's rather disconsolate to admit that 65 years after this official warning, we are facing a possible pre-post antibiotic era in which, according to the World Health Organization, simple infections could once again kill. And in fact, we are here today because unfortunately, antimicrobial resistance amongst pathogens has been recognized as a major public health threat. Therefore, finding alternatives to antimicrobials has a critical importance due to the rise in antimicrobial resistance associated with animal and therefore poultry production. During our research project, we are trying to assess whether peracetic acid used as a wide-spectrum biocide could be a valid substitution for antimicrobial therapy and therefore non-antimicrobial modulation of gut microbiota, which could also interact with performance. The project extends across different years and through planning different animal trials, 2 of which have already been carried out. Amongst other things, we are also trying to understand whether the administration route of PAA has an impact on what is the level of gut microbiota and performance. Therefore, we have already carried out a water trial in which we had administered the peracetic acid through water, and a feed trial in which, on the other hand, we had administered PAA through feed. We have very encouraging results from our first water trial and preliminary results from our first feed trial, which point towards potential peracetic acid applications. For these reasons, I would like today to talk about our first water trial, whose encouraging results laid a foundation for a current feed trial and a pending large-scale trial, and also application in winner pigs, as we will talk about in more details later on. The main aim of the water trial was to assess the impact of 5 levels of inclusion of peracetic acid in administered through water on both gut microbiota levels throughout different gut locations and growth performance. Peracetic acid was delivered throughout the gut thanks to hydrolysis of unencapsulated precursor sodium percarbonate and tetraacetylethylenediamine produced by AGA Nanotech. A total of 96 birds were used during the trial, which were individually wing tagged at day 1. The trial lasted for 14 days and followed a randomized block design in which 6 treatments were administered during the last week. So we had a pre-treatment and treatment week. In total, we had 2 rooms with 12 pens per room, 4 birds per pen, and 4 pens per levels of inclusion. At day 14, after post-mortem, we collected the content of crop, jejunum, and zika, which were immediately snap frozen on dry ice and then stored at -80 until further analysis. Absolute qPCR quantification through linear plasmid standard curve method, targeting the V3 region of the 16S RNA gene, was used to calculate the bacterial cell per gram of sample through the formulae showed on the screen. Moreover, for performance analysis at the intervals showed on the slide, we measured body weight gain, feed intake, water intake, and feed conversion ratio, which are measures for how good the birds performed. Statistical analysis was carried out through fitting a linear mixed model, as you can see in the figure, with fixed effect being the treatment and time for longitudinal data, a random effect being the hierarchy of room, block, pen, and bird when possible. The model, moreover, was fitted through random intercept. And here you can see the results of our analysis for gut bacterial concentration, in which, as I said, we had measured abundance of total bacterial number, and therefore at this point so far is not a qualitative analysis, which is though planned for the next future. So here you can see in different colors the total bacterial number in log 10 per different gut locations. When in red we see the Zika bacterial content, in blue we see the crop bacterial content, and in green we have the jejunum bacterial content throughout the different levels of inclusion from 0 to 50 ppm. As you can see, we found that there was a significant correlation between the treatments and the log 10 bacterial per gram in the crop, especially through a contrast analysis of the linear mixed model. We found out that levels of inclusion at 10, at 50 ppm were significantly correlated to a decrease of log bacterial number, whereas in levels of inclusion at 20 and 40 ppm had a trend of decreasing log bacterial number in the crop. Passing on the performance data, here you will see the results for the cumulative body weight at day 7, day 10, and day 14, with the different levels of inclusion represented by box plots with different colors. Now, a recurring pattern that you will see throughout the presentation of performance data is that we had observed an adaptation period in the first part of the experimental week, in which we observed no significant differences between the treatments, but we did observe some interesting results at the very last part of the experimental week. This adaptation period to the particles from the bird suggested that we might have observed bigger differences if we went ahead with the trial, which laid the foundation for a pending larger scale trial, which will last more than 14 days, and which is planned for next autumn. Now, as you can see here, thanks to a contrast analysis of the linear mixed model, we observed that birds administered 10 and 40 ppm of peracetic acid had a higher body weight, statistically significant compared to the control at 0 ppm at day 14. Very interestingly, we also found out that the body weight gain of the birds associated with a 20 ppm level of inclusion was higher than when compared to the 0 ppm as a control. And here you can see the results from the entire last week, so the treatment week, in which we found out that birds administered 40 ppm of PAA were performing better because they had a higher body weight gain than compared to birds on the control. And this was statistically significant as well. We also found out that from day 10 to day 14, birds associated to 20 and 40 ppm levels of inclusion were eating more than birds associated with 0 ppm control of peracetic acid. So up to this point, we had very interesting results, which were telling us that peracetic acid administered in water for the last week of the trial had an effect on upper gut bacteria and also on performance. So at that point, the natural question was, is there a correlation between this decreased number of bacteria in the crop and performance? So we started doing a few analyses, and the question was, is body weight gain associated to that reduction of crop bacteria that we had seen? Therefore, we had used body weight gain as a dependent variable and the crop bacterial number as an independent variable to fit our linear mixed model. And very interestingly, the model itself was highly significant. Therefore, there was a correlation between the decreased level of bacteria in the crop and the increased performance. In fact, you can notice here that higher level of bacterial number are associated with lower performance, whereas when we reduce that concentration of bacteria in the crop, the performance increase. So to summarize, we had found out that 20 ppm and 40 ppm of peracetic acid administered in water showed a trend to increase the body weight gain. Peracetic acid at both 10 and 50 ppm were significantly associated with lower amount of bacteria per gram in the crop content, whilst 20 and 40 ppm had a trend of reducing bacterial abundance. Very interestingly, body weight gain was significantly correlated to a decreased number of bacteria in the crop. Well, these were very encouraging results, and as I was pre-announcing, we had already carried out a feed trial, during which preliminary results also show that there is an interesting correlation between in-feed administration of peracetic acid and performance. These encouraging results so far demonstrate possible peracetic acid applications in chickens and in species beyond chickens, such as, for example, winner pigs. This also suggests that peracetic acid has a potential use that will possibly decrease the insurgence of antimicrobial resistance, which can ultimately lead to a decrease of environmental antimicrobial resistance. So to conclude, our small-scale study showed the potential use of peracetic acid in water in reducing the total number of bacteria in the upper gut and especially in the crop. However, this link between both quantitative and qualitative measures of upper gut microbiota and amelioration of performance through antimicrobial-like mechanisms is often underlooked. Nevertheless, our small-scale study lies the foundation for further investigation of this correlation. And in fact, we might expect similar results in other species, which is exactly what our pending future project, recently funded, is going to explore in winter pigs. Therefore, we will have certainly more results on this front in the near future. I would like to thank my project partners, AGA Nanotech and Gamma Healthcare, that produced the particles that you had seen during the presentation to achieve peracetic acid throughout the gut, CL and Innovate UK. And with that, I would also like to thank you for your attention, welcoming all the questions that you might have. Thank you very much.
Hello, everyone. My name is Jasmeet Kaler, and I'm a professor of epidemiology and precision livestock informatics at the Vet School, Nottingham. Today, I'm going to talk a little bit about, give you an overview of some of the technologies we are utilizing for early disease detection and some of the ongoing research at Nottingham. I think I'm going to start by maybe talking a little bit about the current state of art in terms of monitoring livestock health and welfare. And I mentioned here current changing state of art because we are in very exciting times with a lot of technology available to us. But how we generally monitor livestock health and welfare is we mostly utilize visual observations, and we know there is lack of data on farms. And how that impacts us is that our understanding of disease is reliant on reporting and recording of that manually collected information. And then one has to question around sensitivity and validity of the predictive models that we build. Given such lack of data, can we really predict and prevent? Change in behavior is often an early indicator of disease. And there is so much rich information in these behavioral signs that can really help us for early disease detection. It's not only this helps us in disease detection, but it also have in it rich information and indicators for positive animal welfare, such as things like play behavior. And thinking about how we can utilize and monitor all such information with all the revolution in technology, this all comes under a precision livestock farming. This term was coined nearly a decade ago, and precision livestock farming is management of livestock by continuous and automated real-time monitoring of various indicators of production, reproduction, health and welfare, and also utilizing that information to look at different types of outcomes and in impact. How does then this helps us? This helps us, all that information that we are recording, monitoring, helps us in early disease detection and hence reducing antibiotic use. All the data that we gather also help us to build better predictive models, and we can come up with preventative strategies, it helps us enhance the transparency of supply chain. And as I mentioned earlier, it also helps us to quantify some other indicators of positive welfare and look at welfare much more holistically. By using all that data and availability of such data in richness of that data, both in terms of variety and volume, also helps us to benchmark and monitor farms, animals on a continuous basis. And then, of course, we can link, this could be linked to payments, etc. But how is that possible? This is possible because of the technological advances that we had over the past decade. The price of sensors is dropping, and it's not only the price, they are much more smaller in size that help us utilize much more widely in various use cases. Not only the hardware or the technology itself has advanced, but also the computational power allow us to then utilize the data generated by these sensors in building algorithms by various techniques such as machine learning, deep learning, and artificial intelligence. So let's talk about some of the example where we've been utilizing these sensors. So the first example is sort of utilizing a sensor for automatic lameness detection. And this is the work we did with various industry partners. So why lameness? Lameness is one of the biggest health and welfare issue in UK and around the world. The prevalence is around 5 to 10%, enormous cost to the industry. And the research we did identified the best practice, which is a long-acting antibiotic and a topical treatment. But the best practice relies on rapid treatment. And sheep are prey animals, and then they mask lameness. So here is somewhere where a technology could really help us to early identify these lame sheep, and hence can help with effective treatment, reducing antibiotic use. And if we look across, catching, treating early is not happening very often. It's only 25% of farmers do say they catch and treat lame sheep early. So this is a perfect use case in utilizing a technology to build something. And what we did, we utilized an accelerometer and a gyroscope sensor, a very small in size. We utilized an ear, we utilized the collar, and we used these signals to develop an algorithm that would then classify a lame versus a non-lame sheep. And you see that as a machine learning task, this was quite nice, because for lame sheep you see a typical head nodding, and we basically just identified that kind of signal and then classified. What you see on this in this graph is sort of 3 different cases where you have sort of walking, standing, and lying, and you can see that the acceleration signals are very different between these behaviors. It's very much same as we were with Fitbit, and it helps us to then look for different behaviors that we perform. But one of the other things we thought about is as we develop algorithms, how do we create algorithms that can stay for longer or adapt to changing environment? So we developed this idea of lifelong learning. So we have a sensor, we have an algorithm, but it not only utilizes the information that it has been trained on, but then also utilizes the information for the current environment. So it's a continuous learning algorithm that updates as it gathers more information. Sometimes just one sensor is not enough, or this is not something that we are interested in. We might be interested in having a range of multiple behaviors, and those behaviors could be indicator, help us to detect the disease early, but also help us as an indicator for positive welfare, as I mentioned, for example, play behavior. So we look at for ill health in calves, and we were interested especially in early detecting pneumonia. And we know that sometimes change in single behavior is not enough. So here we had an array of behaviors that we wanted a sensor to classify, such as locomotor play, which is an indicator for positive welfare, and then other sort of range of behaviors. So you can see specifically in this clip where you can have calves displaying sort of locomotor play, and we had a sensor that was able to classify this behavior alongside with other behaviors with very, very high accuracy. So this is basically a matrix where you have all the different behaviors, just utilizing a single sensor in a collar with accelerometer and a gyroscope, and we were able to classify these different behaviors with quite high accuracy. But it's not only activity sometimes sensors can give us, sensor can also give us sort of location. And then you can see in this particular clip, these are different dots, and these dots are all calves, and they are in real time telling us where the calf is, who the calf is next to, and their movement and trajectory. And this helps us to then calculate sort of various measure of space use and also build social networks. And they can be very useful indicator for early disease detection, as well as with social networks, we can also learn much more about transmission and disease transmission. So on the right-hand side, you can see sort of a trajectory of a calf in a day. So of course, that calf was pretty busy during that day, but we can learn a lot from their behavior and then use novel indicators. Another type of sensor is bolus we are utilizing. So these are reticle rumen temperature bolus we utilized both in cows and in calves. These give us not only the temperature signal, which we can use to see the change in temperature for early disease detection, but what we also use that temperature signal is to build an algorithm for drinking behavior. So the animal has bolus inside, and as the animal drinks, you see these drops, and our algorithm can detect these drops and then drive a drinking behavior. So we can look at the change in drinking behavior, both for disease detection, but also as a welfare measure. Then I'm going to talk a little bit about use of imaging. We are using different cameras, and here is a use of thermal cameras. So body condition score, as we know, is such an important management tool on farms, and body condition score is done visually, and there is also a lot of subjectivity in it. So here we developed an algorithm utilizing a thermal camera. What you see on the right hand side is like a dial, and we have a camera where we build an algorithm where it would look at a cow and then automatically, by utilizing the images and certain feature, classify its body condition score. So you can see sort of the dial that comes here. It tells 3 for each of the cow, then it automatically gives a body condition score. And with this algorithm, we were able to achieve quite a high accuracy. And another use of imaging that we utilized was pyrexia in calves. So here we utilize again thermal imaging. Here the calves comes in the feeder. We developed an algorithm that detects eyes of the cow, and from the eyes we extract the temperature. Especially with COVID, I think we also sort of utilization of thermal imaging or for temperature, and this is a similar principle where we are able to then extract that information for early disease detection. But another very exciting piece of research we are doing is now we have sensor information and the information from images, and we are utilizing and fusing all the different features to create novel phenotypes. And then these novel phenotypes, we are quantifying the individual variation, and these are all the different calves. By combining different information, they are aligned. We have come up with a behavior type and their predictability. And then we are using those novel phenotypes for disease prediction. I think it's quite important to mention technology on farms and technology adoption. And we are also doing some research in what factors influence technology adoption, what it means for farming, how can we enhance technology adoption. But I think one of the key thing of technology adoption is to work with stakeholders right from the start when coming up with different technologies. So we've just been funded a project. It's a large consortium called Decide with various European partners. And what we are doing is we are creating a living lab for precision technologies. By living lab, it means is that it has an elements of co-creation with different users. We use different methods to co-create, and then the user engagement is from the start in the design. So I think it's quite important if we want technologies to be widely adopted on farms and to be utilized in the way that we intend to. So here we're going to have the pilot implementation, and then we're going to evaluate different decision support tools. Finally, I'd just like to say thank you to the group. So I lead a ruminant population health group at Nottingham, and a lot of researcher, and this is a very collaborative work with various industry partners as well as various researchers at Nottingham. So I look forward to answering your question in the panel. Thanks very much for your time.
Good afternoon, everybody. Well, I hope you enjoyed that session. 5 fantastic talks. We got the opportunity now to question those guilty of those talks. So I'm joined by with Salvatore, with Conrad, with Fiona, with Henry, and with Jasmeet. So we are now ready to talk. Our first question in is from Caleb. And Caleb asks, is there a danger of losing momentum from stewardship? Considering Brexit and COVID, how can we encourage investment? So I don't think that's aimed at anyone in particular. I think that's for everybody. So does anyone want to have a stab at that one?
There's always a danger. There's always a danger of lack of momentum. But that's really why it involves every single one of us, doesn't it? Whatever our profession, whatever place we are, is encouraging our colleagues to recognise the significance and importance and our own responsibilities, I suppose. I
think I agree with you there, Fiona. That's one thing I picked up from your talk, was actually that inclusivity comment. It's one thing when I give talks, I say quite frequently that it's not about the vets or the medics. You know, we have a problem, and it's all of us need to sort it out and face it, whether we're scientists, whether we're clinicians, whatever we are. And I think that's a really important thing. So whatever COVID or Brexit or anything else throws at us, that we need to deal with it. And COVID has been a particular problem because all the advice that has been given about not treating blindly has obviously, for reasons which are entirely understandable, has gone out the window during COVID. So now we are facing in the human world an increase in resistance to some extent, and no doubt that problem will filter through. So I think that's one thing that, regardless of the cause, it's a problem we're going to have to face. So.
I mean, I think what's interesting, particularly with regard to the livestock sector, is how different actors and different groups are moving into the stewardship debate. And I think the momentum can quite easily be picked up as we move towards more sort of food chain actors coming into stewardship. I mean, I talked a little bit about the red tractor issue, but we know that supermarkets are also issued about sourcing from farms that are using less antimicrobial medicines and so on. I think how veterinarians are working together to look at different forms of benchmarking with farmers. So I think what we're seeing is a sort of multiplicity of different actors coming into this debate, which hopefully will keep the momentum developing forward as we move on.
Anyone got anything else to add to that, or shall I move on to the next question?
I think one other thing, if I may add.
Yeah, please do.
As well, I think we talked about sort of role of different actors. I think what we are also seeing is advocacy. So there is much more advocacy around sort of responsible use from those actors, not only in participation. The concept of One Health, especially, I mean, I think COVID has brought, and there are more different initiatives around One Health, which are also helpful. And I think finally, kind of linking back to my talk around sort of advances in technology, and I think what we have seen that would also allow to keep this debate, but also enable certain elements that would help us to act in responsible use.
Absolutely. Thank you for that. Yeah, I think that makes a lot of sense, doesn't it?
If I may, I was just thinking this is one of the situations in which needs will create opportunities, and therefore I think that we will face different challenges in the future that go beyond COVID and Brexit. But I think the important thing to remember is that we do have a problem, and we, as Fiona was also saying, the networking and collaboration between what we are trying to put there in the open and innovation, as Jasmeet was saying, is the key for us to go ahead with the work as planned.
Absolutely, absolutely. Right, okay, I'll move on to the next question then. This one's probably aimed pretty much directly at Conrad, but this is CRISPR-Cas a one-shot approach in terms of being an application, or does it need to be applied multiple times during the treatment regime? And that's a question from Mark.
Is this with respect to therapeutic applications or with respect to prophylactic microbiome engineering?
It doesn't specify in the question, but perhaps if you could address both, if you don't mind.
I think, you know, the truth is we don't know yet because we're still at an early stage of R&D. In the study that I talked about in the mouse model, we'd only done it once. I imagine that it would be a repeated dosing in order to increase the efficiency of delivery with regards to the therapeutic applications. But then equally, there are repeated dosings of the antibiotics. So I imagine that since this is essentially a companion biological therapeutic, it would be delivered shortly before or together with the antibiotic. With regard to the prophylactic applications, it in principle could be a one-off in the sense of clearing the microbiome, the gut microbiome of antibiotic resistance genes, but we know also that the gut microbiome is a dynamic ecosystem, and presumably new antibiotic resistance can adventitiously arrive. And so I think that equally one would need to consider screening by sequencing, screening the gut microbiome. I think the diagnostics is improving. Oxford Nanopore have a solution for rapid screening of bacteria. And one would therefore want to do repeat deliveries. How stable you make it, I think, would be determined by your choice of gut commensal. If you were to use something that's endogenous to your host, and for example, we're thinking of doing just that in our forthcoming pig gut microbiome studies, I think we'd want to use an endogenous pig gut commensal as our donor and introduce it. And the assumption would be that since it's come from the pig, it should be stable in the pig. What we don't know, of course, is how much variation there is in the gut microbiomes of different pigs in different parts of the country or world. And so there's still a lot to find out.
Absolutely, absolutely. I mean, do you think there's any likelihood, because obviously we're disrupting, yes, it's a gene, but that gene might well have some linkage to the organism's capability of being dominant within a particular environment. Have we got any evidence that by disrupting the target organism, we're actually going to disrupt the rest of the microbiome, or do we think that's relatively stable?
Well, that's a good question, and we don't know, but knocking out an antibiotic resistance gene in the absence of antibiotic selection shouldn't change the balance of the microbiome. Obviously, when you then administer an antibiotic, you're going to select for antibiotic resistance genes, and if they're present or dormant, then they might come into the fore.
Yeah, absolutely. Excellent. Thank you very much. Got a question from Tamsin, which is possibly related to a number of you, actually. It says, what happens if plasmid-bearing commensal organisms are introduced into the non-cleared gut environment?
I'll have a stab. I'm not sure I understand the question in the sense that plasmid-borne depends on the plasmid and whether the plasmid has provides a selective advantage to the bacterium that carries it. You know, in our case, we'd be introducing a conjugate of plasmid that carries the CRISPR-Cas9 machinery. That's going to inactivate antibiotic resistance genes. It might also be used to inactivate virulence genes. that comes back to your question: how does that change the fitness? And it's going to depend very much on the selection pressure.
Yeah, I think Tamsin's question very much depends on what is on the plasmid in the first place. Is it another AMR gene? Is it another survival gene? But it could have an effect, I guess. But that's another one we'll have to see as time moves forward. Just to encourage the audience, please do use the blue ask button to ask a question. We're a lovely bunch. We will answer it if we can. And Fiona has done exactly that. No, it's a question for Fiona. I beg your pardon. I can't read. So question for Fiona, not from Fiona. And it's a question from Jude. And it's, what are the biggest wins in assessing medicine's use, reducing, refining, replacing antibiotics?
What are the biggest wins? Well, I think currently certainly in the ruminant sector, which is probably the most challenging for collecting data, is actually to be able to evidence what antibiotics we're using and how to evidence that use on a wide scale. We've got a medicine hub that is up and running, but as yet we don't have a lot of farmers inputting data into that. So that is a big win, because as you'll have heard said many times, we can't manage where we are until we've got it well measured. And engaging the vets in that is, I think we've got 70,000 beef and sheep farmers, lots and lots of small businesses dealt with by general practitioners, and who are themselves often in small businesses as well. So that's really just engaging hearts and minds, and everyone seeing their own responsibility and getting on with their little patch is the key, I think.
Yeah, absolutely. In the small amount of research I've done on AMR in agriculture, that's exactly the situation. And it's linked to the next question, actually, which is from Renee, that says, are there any annual livestock AMR surveillance schemes in the UK? Any possibility of government funding for this? And I think that's probably quite a pertinent follow-on question, given what you've just said.
Well, there's surveillance going on. Currently, a lot of our data is at the level of national sales. So we got data nationally, not on AMR so much as actual usage. There is AMR stuff done at kind of meat shelf level, as it were, in supermarkets. I suppose it's tackling it at all levels, isn't it? Is a government funding appropriate? There's some and some. I think people taking their own responsibility is equally as important.
Thank you for that, Fiona. Anyone got anything to add to that, or
can I?
Yes. Oh, Sal first, and then we'll go to Henry.
Oh, thanks. No, I was just going to follow up on that, saying that despite of not having a real probably structure in terms of national surveillance, there are a lot of groups that are actually doing that on different environments. For example, either water, river, or animal-based, so livestock, AMR spread. So I guess that just follow up on what Fiona said, maybe putting all this data together in a database can constitute that information that probably at the moment is a bit lacking.
Yeah, develop a hub to bring it all together.
Yes.
Yeah, it makes a lot of sense.
In the UK, we've got the Veterinary Medicines Directorate sort of annual reviews of antimicrobial sales in veterinary medicine, taken from prescription data, which has been a very useful resource for us in research over the last few years. And you know, there's been some really interesting trends, and they produce their very good reports on the different species types and so on, which has been great. But I think what's also interesting is the way in which, coming back to these different actors, that they are themselves monitoring more and more use. So you've got veterinary companies which are very closely monitoring prescription use in the livestock sector. You've got supermarkets now that are monitoring the use of antimicrobials in their suppliers. So I think there's a sort of multiplicity of different data sets that's emerging, as is often the case when an issue comes up. What's the challenge is to bring these together and to share the data. I think, you know, the trouble is with data sets is they very quickly become privatized, and not everyone can get access to them. And that's where the problems arise, I think.
I think you're right. And I think there's an information piece to be done around this as well, because there does come a point where we can't use any less antimicrobial than we're using, because as we were saying earlier on, there's a welfare issue, and we've got to make sure that the animals are properly cared for, and that's just not even a debate, that's just a fact.
But the rhetoric's really important there, because if we talk just about reducing, then we lose that. The responsible use is what is so key.
Absolutely.
I think. And as we, the way we talk about it's really important.
Just wanted to add, I think we talked about the usage data. I think perhaps thinking about the AMR surveillance in UK and what data we have on that. And I think it's we do have the clinical sort of from the data from the clinical isolates, but we don't have sort of from healthy animals. So I think we also need to realize sort of the patterns that we see in the report. How do we interpret them, and what do they mean, etc. as well.
Got another question from Kaler. This is actually for you, Jasmeet. How easy is it to implement a monitoring system on a different farm once it's set up, like the one, I think it's saying like the one at Sutton Bonington?
Yeah. Okay. So I think it's around sort of how generalizability of the various technology systems and various innovations that we develop. I think if we look sort of way back, I think my answer would be it's very, very difficult because we lacked in sort of data pipelines or data processing tools. But I think we are getting there. There are a lot of technologies now available that have interoperability and they could be utilized onto the farm. Talking specifically about the system that we have, we at Sutton Bonington, we are at some of our technology is at very early stage of innovation. They are really state of the art, kind of really developing, pushing the boundaries. And I think I would say their technology readiness index is low. So some of the technologies are around 3 or 4. And I think our aim would be to really do enough experimentation and do further trial work on another farms before they move to a readiness level of 7 and 8 and available commercially.
Another one from Caleb. I think Caleb's on a question roll. It says, are there any other biocides you think that could have the same effect and feasibility as PAA? And that's obviously to Sal. So,
yes, potentially yes. There are, although it's very early stages, and therefore it's potentially a genetic biocide. And therefore, more or less on the same line of the conversation we had about the plasmid, it really depends on the downstream effect that you have on the community. And therefore, once you have established what kind of levels are functional and what kind of effect you aim to have, then you can also plan what are the other application of it.
Okay, excellent, thank you. And the other question from Caleb is for everybody, and it says the ANIR report is cited in most AMR meetings. How long can we rely on that report before it becomes out of date? That's a good question.
I guess that this also is a follow-up question on the data set we were talking about, because ideally, I think that having a real-time report every year, that would be the best way to tackle the problem. But we had also said that we lack in a kind of surveillance at the moment. So I will say as frequently as we could, but and the situation we know is changing, and we know we are creating either more resistance or there are environments in which there is a selection towards determinate genes. So I will say that we will need the report quite soon, but probably a frequent monitoring of the situation is the key.
And certainly the rumour reports, so 2016 to 2021, reporting on where people have got to within the different livestock sectors and setting the new targets, and then they're committed to reporting on an annual basis. So maybe not quite so widely cited, but really worth turning to for the most up-to-date status, as it were.
Absolutely, thank you for that, Fiona. Anyone else got anything to add to that one? Oh yes, Henry, sorry, he's got your hand up.
No,
Sorry, sir.
No, I mean, it was interesting. Jim O'Neill gave a talk sort of updating the report to a conference in Bristol last year, which was actually very good, and he was able to sort of bring it forward a bit, which was very interesting. But as I think I said it in the paper that was presented here from myself, I think that the two things that have sort of gone on a bit since the O'Neill report. I mean, I think, as I said in that paper, the emphasis on rapid diagnostics in livestock, hasn't really come to fruition in the extent perhaps to which people thought it might at the time, for reasons that we explored in that paper. And I think, as was the time also in the middle of the last decade, there was this great emphasis on behavioral change as the driver for changing things. And again, I think that emphasis was partly misplaced. And I think more and more we're becoming much more attuned to the value of more holistic approaches to whole farm environmental change. Behavioral change can, as that horrible phrase, grabs the low-hanging fruit, but it's the higher hanging fruit that we can only really change through fundamental infrastructural and structural change, different monitoring types of surveillance and monitoring data, bigger things that actually the O'Neill report didn't really look at. So I think that's where we've come forward a bit since the mid-1990s and 2000s.
Thank you for that, Henry. That's an excellent point made there, I think. Thank you. Just looking at the time, we've probably got time for this one last question. And this is for several of you, I think. This is from Mark, and it says, besides accuracy, which other factors are important in pen-side point of care testing, such as duration, cost, ease of use, etc.?
Well, I could say a few things simply because that was kind of what we looked at. I think, I mean, obviously accuracy is a big issue and I'm talking about veterinarians here, not medical practitioners. But I think there are all sorts of issues associated with the farm environment. The nature of cross-contamination, the difficulty of using some of these things, which has hindered, I think, their adoption more widely within veterinary practice. I think the other thing I think is that some of the sort of veterinary laboratories are getting very fast at getting stuff back to veterinarians in the field with a smartphone, and so the whole essence of rapid can only take place at the side of the animal is also being challenged within the veterinary side. So I think all sorts of things are developing very fast in that area. The difficulty, of course, is bringing them to market, but that's another issue. Excellent,
thank you. Anonymous asks, do we need a new O'Neill report to set a new direction? Well, I think our speakers have largely said, I think we probably sort of do. But anyone got any, and I think this will need to be our final comments. Any final comments from the panel based around that question? And that'll be a no. So I think we've probably covered that in the comments that we've made, and I think, yes, we probably do need to look at that again because things have changed in a very positive way. Groups like RUMA and various other groups have done a lot of work in helping the MIG per kg values get down to somewhere that is still useful and responsible and sensible for the treatment of animals, but below where they once were. So I think we're moving in the right direction. We probably do need to regroup, and we can perhaps use some of the innovations that we've heard about in the talks that have been so excellently produced by our speakers today. So I think this is an opportune moment to close this part of the meeting. I'd like to massively thank Sal, Jasmeet, Conrad, Fiona, and of course Henry for all their contributions. Brilliant talks, thank you for those, and a brilliant conversation at the end. With that, I'd like to thank the audience for sending in their questions and attending. And thank you for coming to this joint RUMA and SFAM conference. I bid you all a safe trip to your other room or wherever you're going. So,
thank you.
Hope to see you in the flesh soon.
Thanks for having us.
Bye, everyone.
Thank you.
Thanks.
Thank you.